Welcome back, friends. So, we are moving into the next part of culture media. So, in the previous discussions, we have seen the different types of media or the classification of media based on various criteria. And now we will discuss the remaining classes, especially the transport media, the anaerobic media, the sugar media, and also the indicator media. So these are the four types of media we will address in this part of presentation, right? And I hope you can follow the discussions and you can have your comments in the comment box if you have any doubts. Transport media, actually this one is not a nutrient media. It's not designed at all for cultivation of microorganism, but for providing a protective type of media which is used for the transportation of a specimen from the site of sample collection to the laboratory without affecting the organism's features, right? So the two important things what a transport medium do is after the collection of a bacteria, collection of a sample, they prevent their changes. That means they protect their biochemical or other type of changes, right? That means they help them to survive in short. The second thing what a transport media do is a transport medium will prevent overgrowth of non-pathogens or non-desired organisms during the or in the time taken for the transportation of this sample from the site of collection to the laboratory, right? So these are the two important features what a transport medium do, right? So by definition it is a media, or in fact it's a special media formulated to preserve a specimen and minimize bacterial overgrowth from the time actually it's from the site of collection. It's not time, it's wrongly typed. From the site of collection to the site it is received at the laboratory to be processed. Okay, even if it is time, you will get a meaning, right? So you can see certain examples in the next slide. Yeah, Stewart medium, buffered glucose, glycerol saline, via medium, carrion blay medium, amines charcoal medium. All these are different media for bacterial transportation, right? You can see Stewart medium is a non-nutrient soft agar gel which contain a reducing agent. This reducing agent will prevent the growth of other organisms and will provide a protection. Used for transportation of Neisseria gonorrhea, Streptococci, Enterobacteriaceae, and certain fastidious organisms, and even anaerobic organisms, since it's having a reducing agent. You can see the composition over there. Right, and the second example is buffered glycerol saline. It's for Shigella species. Then the third one is via medium or Venkatraman Ramakrishnan media. For the Vibrio cholerae again, which consists of crude sea salt and peptone as the ingredient. You can note the point that there is in most of these media there is no nutrient substance, only certain minerals and other things are provided. No any nutrients like glucose or peptone are provided because it's a non-nutritive preparation. Then another example is carrion blay medium. It's a buffered solution of sodium chloride, sodium thioglycolate, disodium phosphate, and calcium chloride. It's a semi-solid one used for the transportation of Vibrio cholerae, Shigella, Salmonella, etc. Then amines charcoal media for fastidious organism. And of course, this one also you might have heard, VTM, viral transport medium. In this time, we are familiar with this coronavirus, and for after collection of the sample, the swab is taken, and this swab is actually put in the VTM, viral transport medium, and then that is transported into the laboratories for the processing. You can see these examples. These are transport media for bacteria, different bacteria. You can see all these are amines transport media, Then you can see Carrie Blair media. All these are different examples. Okay, now the second class is anaerobic media. Anaerobic media, of course, is used for the cultivation of anaerobic bacteria. There are different anaerobes. For example, you can recollect examples like Clostridium is one of the very relevant anaerobic organism which cause tetanus and other type of food poisoning. Then these organism has to be cultivated in anaerobic media. Right, we know how or why these organisms are unable to grow in anaerobic media and what is oxygen toxicity. All those things already you might know. So here is an example for anaerobic media. That's very common one is Robertson cooked meat medium, RCM. Right, it's otherwise also known as cooked meat broth or CMP, which consists of a fat-free minced cooked meat of ox heart, and that's why it's called cooked meat. And also it's a nutrient broth. In fact, it consists of this cooked meat. It supports the growth of both spore-forming and non-spore-forming anaerobes, which are obligate in their requirement. Right, and as a procedure, before inoculation, the medium has to be boiled to make it oxygen free. While you boil, the oxygen will get released out from the medium, and then we'll tightly pack this one and will be used further. Then the media which contains certain chemical contents like glutathione and cysteine, which help in removing any residual oxygen. Right, and these reducing agents will exert maximum activity when in the cooked stage. Right, that means the glutathione and cysteine are present in the meat. When they are cooked, they have maximum activity to consume the already present residual oxygen. Then also there is unsaturated fatty acid in the meat will consume oxygen if it is there. Then dextrose in the media will promote growth of anaerobic organism. Right, so all these are the different features. Then after inoculation usually is covered with a layer of sterile liquid paraffin oil to prevent the entry of oxygen into the medium, so that the external entry of oxygen is prevented. So in this way this becomes an anaerobic medium. Clear? Here is another example for anaerobic media, thioglycolate broth, which contains sodium thioglycolate as a reducing agent. Right, which maintain a low oxygen tension by removing molecular oxygen from the environment of this media. Right, so it creates an anaerobic condition and it's actually used for differential for determining the differential oxygen requirement by the bacteria, and you can categorize organisms based on the oxygen requirement as obligate, anaerobe, aerobe, facultative anaerobe, like that. Right. Now the next category of media is indicator media. So indicator media are those which contain an indicator that changes the color when a particular kind of bacteria grows on them. We have already seen certain examples for that. Mekong agar is an example, lead agar is an example, EMB is an example. That means all those contain an indicator, right? And that means most of the selective differential or selective differential media which contain indicators, right? Then here is a media which contain an indicator. Christensen's urease agar consists of phenol red as a pH indicator. Of course, you know what a pH indicator do by the examples of the already we discussed in MCA and EMP. Then urea present in the media will be hydrolyzed to release ammonia, which actually we know ammonia will change the pH into alkaline condition. And this pH detected, shift in pH will be detected by phenol red and will turn the media from light orange to pink color or from yellow to pink color. Now finally the sugar media which contain any fermentable sugar, right? So there will be examples like glucose, lactose, fructose, sucrose, whatever it is. We have a fermentable sugar in that media, usually at a concentration of 1%, right? And occasionally there will be a pH indicator also added to detect the acid production, and the gas production will be detected by providing a tube called Durham's tube, and this is a small tube which we put in the test tube in the inverted position so that any gas production will be detected, right? So there will be pH indicator and also Durham's tube to detect the fermentation of the particular carbohydrate, right? So this is about the sugar media. So by that we are coming to an end of this part of culture media, which are widely used in microbiology laboratory. You can have a review of yourself: what is nutrient agar, what is nutrient broth, what is EMB, McConkie agar, endo agar, blood agar, chocolate agar, Stewart's medium, Venkataraman Ramakrishnan medium, thioglycolate medium, Robertson cooked meat agar. So all these are different examples. You have to be familiar with all those. You can also connect with different classes of media through these examples. Fine. So hope you could understood all those.